In this video, we'll be talking about things moving across the plasma membrane. Now, the plasma membrane, of course, is separating the inside of the cell and the outside of the cell. This allows the cell to maintain homeostasis by controlling things entering and leaving. Plus, it's another way for cells to make products and have them leave the cell. So let's look at some of the basic ways. Now, the first thing to really think about is that the lipid bilayer that we talked about in previous videos is selectively permeable. And what does this mean? It means that only certain molecules can cross. So some things are allowed to cross and some things are not allowed to cross. Now, substances may cross the plasma membrane in two major ways, and then there's subtypes of ways within each of these two major categories. And the two big ways we can break all these mechanisms back down is whether they are a form of passive transport. And passive transport means I don't have to spend energy. And others use active transport, where I have to invest energy, and usually that energy is in the form of things like ATP. So all the mechanisms we're going to see in this chapter either fall into a passive or an active category. So let's start talking about the variables that determine how something gets across. Now, I want to point out some things are never able to cross, but three variables determine how you cross, whether it's by active or by passive. So, one of the big things is the type of substance. That has a big role. Your chemistry, your shape, your size. The other thing is the plasma membrane's permeability to the substance. Now, remember, this can vary from cell type to cell type. We got to always remember that not every cell is the same in terms of the receptors it has on it, what can cross the membrane, what the cytosol is like inside, and the list goes on. Also, the concentration of the substance in the cytosol and the extracellular fluid. Because, of course, things move from high concentration to low concentration. That can be a driving force to move things across a membrane if it has the ability to go across. So let's have a look at some of the mechanisms. Let's start with passive transport. And when I'm looking at passive transport, we can really consider two major categories, and that would be diffusion, and the second one is osmosis. Now, under diffusion, we have simple diffusion, and we have facilitated diffusion. So let's have a look at some of those. Now let's talk about diffusion. Diffusion is things moving from an area of high concentration to an area of lower concentration. And concentration gradients is really the force that drives many types of passive transport. The example that's given here is if you put a drop of dye in a clear water. Over time, you'll notice that, if it's red, pink dye here, you notice that it moves and it diffuses from the area of high concentration to areas of lower concentration. So more dye molecules in the fluid on the bottom of the beaker than the top. Now, the difference between the areas where it's really high concentration and areas where it's low concentration, we call the concentration gradient. And this is one of those potential energies that if you go back to and look at the core principles that we talked about in a previous chapter, you'll see that it falls into that category. Now, all molecules have that energy type, which we call kinetic. And also, as long as there's thermal energy is present, we have the ability for random motion of these molecules. And not surprisingly, given enough time, if you randomly allow molecules to move, they'll disperse themselves over an area. Now, here's the example that they're showing you of diffusion, is movement of a solute from, the keywords are high to low concentration. And when things do that, we say that they move down their concentration gradient. So when things move from an area of high concentration to an area of low concentration, it's moved down its concentration gradient. And this only happens until we reach equilibrium. Now, equilibrium does not mean there's no real movement, that everything's frozen at that point. Untrue is that just means there's no net movement. And let's talk a bit about that. So here's the example they use. They have a beaker of water. They put a barrier in here. They put a high concentration of sugar in the water here and a lower concentration. And then what they do is they remove the barrier. And of course, the molecules that are here, they all have random motion. So they're all moving a little bit, right? And just by sheer luck, chance, they will move, and more will start to occupy this side. So we haven't added any energy to the system, but through this random molecular motion, some of the molecules have diffused over here. Now this process will keep going. Movement, a net movement, an overall movement from high concentration to low concentration. And you eventually hit a point that the area over here that originally was high concentration ends up having the same number of molecules as the area here, because a number of the molecules that were here diffused over. Now, as I said, this does not mean that nothing moves. All these molecules can have that energy, and they can be moving a bit. But when we say net, that means the overall effect. Because, even here at equilibrium, maybe this molecule will end up over here, but there'll be an equal number of molecules that will end up over here. So the net effect is 0. So let's just review quickly. Diffusion is a form of passive transport, and solute molecules move from high to low concentration, and we say they move down their concentration gradient until equilibrium is reached. And remember, equilibrium is no net, no overall movement, right? There's still movement of individual molecules, but the number that move from here to there will be equal to the ones that move from here to there. Okay, let's talk about simple diffusion. Now, simple diffusion is pretty straightforward. It's most non-polar solutes, oxygen, carbon dioxide, lipids, many hydrocarbons, they can pass through the phospholipid bilayer without a membrane protein. So it's just, they have the right chemistry that they can actually just pass through this lipid layer. Because remember, when we look at the phospholipid bilayer, we have a hydrophilic bit and we have a hydrophobic bit. And the chemical nature of these things, these nonpolar molecules, allows them to actually pass through this middle region and come out the other side, or vice versa. And that's simple diffusion, and it's the easiest type. So most nonpolar solutes, and let's just review, oxygen, carbon dioxide, lipids, and hydrocarbons, they are pretty good at doing this. They can pass through the phospholipid bilayer without a membrane protein. So they don't need a protein to grab them, like one of these transmembrane proteins to grab onto it and then move it through. They just kind of walk right through, diffuse right through that phospholipid membrane. But not everything can. It's only if most of the nonpolar solutes can. Okay, facilitated diffusion. The name tells you a lot. Facilitate, to facilitate, is to help, to direct, to ease, etc. So facilitated diffusion. Here, this is how charged, or remember the last time it was non-polar solutes? This is how polar solutes. So ions, glucose, they can cross the phospholipid bilayer with the help of a membrane protein. And often we refer to these as carriers, or a different type would be a channel. And let's have a look. So here's a case of these ions. Okay, these ions just don't have the right chemistry to pass through. They cannot pass through the phospholipid bilayer. So if they want to get into the cell, not going through the cell membrane, at least the phospholipid bit. But what if there are specific carriers that are designed to allow them to pass, or channels, I should say, I should refer to these as channels. If there's channels that allow them to pass, then they can pass through. Okay? And these different channels will be designed for different things to move through this way. So, what passes through the channels? So I said things like ions do it. And we're talking about polar solutes that pass through. Now, there's another type of facilitated diffusion. The first type we saw uses channels to go through. Okay? The second type of facilitated diffusion can use a carrier. And in the case of a carrier, remember, you come in, you bind on, you cause a conformational change, and you release the substance on the other side. And so both of these fall under the category of facilitated diffusion, either using channels or using carriers. And we're going to stop the video there because the next one will be on osmosis.